Good afternoon, everyone. My name is Molly Sullivan, and today I will be presenting on the work I do in the Department of Biological Sciences alongside Dr. Melanie Higgins. We are working to study the biochemical properties of the GH1 enzyme family. So to give a little bit of an overview of this presentation, we're going to start with an introduction into mucins and what GH101 is, and then talk a little bit about our methodology, and then finally a few preliminary findings. So jumping right into mucins, mucins are components of mucal membranes. They're found throughout the human body and in the environment, so they're super prevalent. They're glycoproteins, meaning that they're proteins with different sugars attached. So here is shown a mucin core. It's an amino acid, so typically a serine or a therine, with different sugars attached to it. And those sugar chains are what determine the function. In order to understand mucins, we need to be able to understand those sugar chains. And in order to understand them, we have to be able to remove them, which is where GH101 enzymes come in. GH101s are glycosyl hydrolases in the GH101 101 family, meaning that they are hydrolyzing that bond between the sugar and the amino acid and breaking it, allowing us to remove the sugar and study it separately. So in this picture here, you can see several different mucin cores. So there's a variety of different core structures. And currently, we've been able to find, or they've been able to find, different enzymes or glycosyl hydrolases that can degrade about 3 of those different cores. So there's still a lot out there that we have not yet been able to characterize or remove the sugars from. So our goal is we want to find enzymes that can remove different mucin cores from the mucin protein. So to start, what is the significance of this? Like, why is this important? We are looking at these enzymes that can cleave and isolate the saccharides off of different mucin cores because mucins are so prevalent throughout humans and in the environment that there's a wide variety of things where this type of tool or enzyme would be super helpful. It would allow us to characterize the function and behavior of different mucins, which has a lot of different applications in disease and drug research, specifically in cystic fibrosis and mucinous carcinomas, as these have a lot of mucins involved in the development of the disease, and also these GH101s could potentially even be helpful in different treatment options. So our main question is, can these GH101 enzymes degrade different core mucin structures? To summarize, because we cannot yet find ways to isolate the glycan from the mucin proteins, we cannot yet identify the full sugars on the proteins to determine their function. So this is the question that we want to try to answer to figure out if we can have different tools unveiled that we can then use. So to go over methodology, to start any of these processes, we need to first be able to purify those proteins, so our GH101 proteins. And then once we have that protein solubilized and purified, we can then study its biochemical aspects and different structural aspects. So to start the protein purification, we first take those GH101's proteins and we clone and transform them and then grow them in E. coli. So then we get something like a pellet, and that pellet is full of bacteria and E. coli and our GH101 proteins. So we want to try to do our first purification step to isolate those GH101 proteins. And to do that, we use something called nickel affinity chromatography, which is shown here in those different resin columns. And those columns have a resin in them. So we first take our pellet and we lyse it. So we cut it up into different bits and hopefully separate the GH101 from the E. coli. And then we can run it through those columns, which filter it. And so the E. coli goes into the flow through, or that little Erlenmeyer flask beneath the column. And then we can collect the other proteins, or hopefully our GH101s, into different elutions or fractions, as we refer to them. So at that point, we've done our first step of purification. We hopefully have our GH101s somewhat isolated, and then we want to analyze those fractions to see if we've been able to actually find what we were looking for. So to analyze those fractions from our first purification step, we run an SDS page gel. And what that does is it separates out the proteins that we found by weight. So we can do something called a prop param analysis, and what that means is we take an online software and we just put in the amino acid sequence of our protein. And it tells us, okay, what is the specific weight of our protein of interest within this protein? So for this particular one, it's GH1013AIF7, and our protein of interest is about 77 kilodaltons. So as you can see here in the gel, there's that one right next to the one where it says 75 kilodaltons. That is our ladder. So that has a variety of different proteins, so it's kind of like our standard, so we can test them against that. And as you can see, elutions 1, 2, and 3 have a lot of protein in that 75, 77 kilodalton range, meaning that we have found our protein of interest in this particular purification process. So at that point, we can then take those 3 elutions that have the protein of interest and concentrate them and then run them through our second purification step. And our second purification step is called our size exclusion chromatography. So that's this machine shown here. It's called our FPLC, or our fast protein liquid chromatography machine. And we can insert the protein into that, and as you can see, there's a really large column. So it's much bigger than our first purification step. And what that does is it further sorts all of our proteins by size and allows us to move into the next stage of experimentation. Okay, so to overview the purification process, we start by protein production in E. coli, move on to lysing the protein, and then we can have our little bits of protein that we can then do our preliminary filtration step, which is the nickel affinity chromatography, and then we can move into analysis of that step using our STS page gel to look at the protein of interest and whether we found the correct weight. And then finally, our second purification step, or our size exclusion chromatography. And then once we have that pure protein, what can we do with it? And that is first the biochemical aspect studies. So we're identifying these proteins and determining how are we identifying these proteins and determining their actual biological activity. We've collected all these proteins, they're soluble, we've been able to isolate them, and now we want to find out what can they do. Are they active in the way that we are looking for? And to do this, we collect those proteins with a variety of different mucin cores and see, and just kind of let them be and see what the results are. So have the cores, and if they have been degraded, what specific cores have been degraded? Where is that biological activity? And that's called biochemical assays. And so if we can determine that some of these proteins do in fact have the ability to remove the sugars from some of the mucin proteins, we then want to ask the question, why is it working that specific way and not with other cores, but this particular mucin protein? And to answer that question, we can look at the structural aspect. So we use crystallography to visualize why an enzyme is working with the substrates it is and how they bind. So we can do crystallography experiments and actually figure out the structure of a protein and then examine the binding sites and where it might be active on the mucin protein. So we can now get into some crystallography and some preliminary results. So we have not yet done any biochemical experimentation on our protein. We have found a fair amount of soluble proteins, and we've been able to start some crystallography experiments from there. So we start this by doing crystal screens, which is not shown here, but it's about a little piece of plastic with about 92 different wells on that, and we can try out different salts or buffers with the proteins to see what conditions would be optimal for crystal growth. And then once we figured out that, we can then do crystal optimization screens, which vary different elements of the buffer or salt, so maybe changing pH or precipitant or concentration to further examine the best possible conditions for crystal growth in the hopes to get a large crystal where we can then examine its structure and how it might be biologically active. And this image just shows the specific method we use, which is the hanging drop method. And so in summary, mucins are prevalent across the human body and environment. And to understand mucins, we need to be able to remove the sugars off the mucin proteins. And in order to do that, we are studying GH101s. And these GH101s might be able to cleave these sugars off of these mucin proteins, and then we would be able to isolate the mucin cores and study them, and then apply that to different areas of research. Any questions?
So with all the proteins you've worked with so far, have all of them been soluble?
So we've actually found a good few that have been. We've had relatively good results in terms of solubility. But again, there's still been several that haven't been, or I'll go through slide, I'll show you here. We've had a couple that have had contaminants, meaning that if there's another kind of protein present near our protein of interest, so it's kind of hard to isolate it specifically. But we have had some good results and been able to do some crystallography experiments with the soluble proteins. But even the ones that are insoluble, it's likely that they don't have much biological activity, because if we can't isolate them, it's a similar mechanism to why they wouldn't be able to function biologically.
How do you identify proteins of interest?
So to identify our specific proteins of interest, I'll go back to our question slide. What we did was we looked at a whole wide variety of amino acid sequences and proteins, and we selected glycosyl hydrolase amino acid sequences that were very unlike what we currently understand and what we currently have biological activity for, in the hope that these proteins that are much farther away from what we currently know might have some new biological activity, and we would be able to cleave different sugars off new cores that we haven't previously identified.
How did you choose the proteins that you ended up looking at?
So as I just said, the proteins that we ended up looking at were the ones that were kind of dissimilar from the ones that we had previously been identified as prevalent.